So before we get into vision specifically and audition specifically, I'd like to talk a little bit about some of the essential sort of aspects of stimuli that we can describe and discuss. One is the modality. When we talk about the modality of the stimulus, we're talking about the type of energy, right? So light, right? It's a form of electromagnetic radiation, and it's a narrow sliver in a larger electromagnetic spectrum that we actually have, specialized photoreceptors that can detect, different, wavelengths within that, narrow spectrum that's correlated with visible light. But that electromagnetic radiation within that range is the modality of light, right? This is the visible light modality. We're talking about audition, right? We're going to be talking about something vibrating like this, generating pressure waves that propagate out in a medium. Most typically air, although you can hear underwater, right? Some organisms do that more often. And that would be, audition. That'd be sound waves would be the modality, right? So within those modalities, light, let's say, or sound, you can also talk about various qualities. Like you can have a soft sound, right? Or a louder sound. You can talk about the amplitude, the intensity, basically of the sound. Within light, that modality, you can have certain qualities, like, for example, the perception of different colors, right? Basically, that are associated with, different wavelengths within that visible light spectrum. And the nervous system kind of codes aspects of modality in two sort of primary ways. Oftentimes it uses what's known as a labeled line code. So that means that there's a very specific type of sensory afferent, sensory neuron, right, that's specialized for the detection of a very precise form or type of stimulus. So we have photoreceptors we're going to see that are localized in the retina that will absorb, wavelengths, and we're going to see that rod cell, that I showed you, doesn't care, which wavelength within the visible light spectrum, strikes the rhodopsin, the photopigment that's found here. It will respond. However, there are also in most of us these cone cells that are another form of photoreceptor, which has more of a cone-like kind of external segment here. That will express different photopigments that will absorb, sort of smaller segments of the entire visible light spectrum. So that can be utilized, to respond to wavelengths that most often correlate with a perception, let's say, of red or green or blue. However, while there are these labeled lines, and we've got them in all sorts of different sensory systems, the nervous system doesn't have like a specialized photoreceptor, cone photoreceptor for fuchsia, or some other kind of color, like every color that you could potentially perceive. So there is also what we call this sort of pattern code. So basically, like many visible light inputs to the eye are going to be composed of multiple wavelengths that will activate, let's say, the red photoreceptor, to this degree, and the green, photoreceptor to this, and the blue to this. So there's a pattern of activity across those labeled lines that will, provide some unique input that can be distributed into networks that can allow for far more, discriminative capacity of, with different colors. So it's a great evolutionary strategy if you think about it, because you're not producing a fuchsia, neuron or I don't know, I'm not a color guy, but all these different colors. You're just producing a few, that require only so many genes, and then the pattern of activity across those labeled lines, allows for the appropriate distribution, to allow for great discriminative capacity in different regions of the cortex. So this is true, for example, in taste as well. You only have so many labeled lines in your tongue. You've got a sweet taste receptor. You've got a salty. You've got a bitter. You've got a sour. Acid. You've got an umami receptor. It seems we have additional ones now that we've identified, a sort of starch receptor and others. So maybe a fat receptor. But there's a limited number of the labeled lines. But it's the pattern of activity across those various labeled lines that allow for the selective distribution of different patterns along different networks that can converge on different columns that could generate and underlie, great perceptual complexity and discriminative capacity. There's also the duration of a stimulus. So that's how long a stimulus lasts, and that's typically encoded by ongoing persistent neural activity. So the nervous system, however, is sort of best at detecting changes in activity. And there are these sort of more top-down kinds of network biases that can exert some control over the distribution and the perceptual experience of inputs, even though a stimulus may still be present and you still be maybe getting some kind of firing, from the specialized receptors, some detection of that stimulus. Because of these, sort of psychological changes, things like habituation to a stimulus, where, if it's a repetitive stimulus, it keeps going, and, it's not a threat, or it's not, somebody poke, over time it may still be getting the touch receptors responding to the poke, but you're not going to get the perception, won't persist to the same degree, over a long period. The same with the flip side of that is sensitization, like, they poke hard or something like that. You may be more attentive. So these sort of central nervous system networks that are involved in attention, and monitoring, and sort of reflection, will also play a role in how long a perceptual experience may persist. The duration of what you're perceiving. But basically, a change, is something that usually is sort of best, detected by our nervous system. Then there's also the intensity, which is, basically how strong a stimulus is, like a light touch versus a hit, right? And that's coded by the nervous system typically in two ways, and it'll depend on the particular system that we're describing, what applies most significantly. But for example, like in the ear for a very loud sound, right? Like very, intense sound, you're going to get neurons that are going to be moved more significantly. These are going to be called hair cells. But they're going to go back and forth faster, and they're going to end up firing action potentials at a much faster frequency. So this is called a frequency code. So the firing rate of the cells that are responsive to the specific stimulus will actually increase their rate of firing. But it turns out that a very intense, that wasn't very intense, but a very intense, loud sound will also cause greater movement of, some of the equipment that we'll be examining in the inner ear, and you'll get more of these cells actually firing. So you're going to get, what's called a population code. A larger number of cells are going to be actually active in response to a more intense stimulus. So both of these could be, at play depending on the system that we're examining. And then finally, there is location. Like, where is a stimulus? How do you know where the stimulus is? And we're going to see that the nervous system loves maps. It loves maps. Like we talked about the homunculus before, right? The map of the body. There's a mapping, what they call a retinotopic map, a map of, the electromagnetic radiation in the range of visible light that's reflecting off of objects in our environment that's arriving in very specific regions of the retina. And that's going to be, that mapping is going to be preserved, and, continue up into, the primary visual cortex in the occipital lobe. So, if I touch here, well, this wire, this particular touch receptor, is going to be, traveling in a very specific part of the cord. It's going to be arriving at a very specific nucleus, in a portion of that nucleus in the brainstem. It's going to be going to a very specific, thalamic nucleus in a very specific part of that. And it's going to be distributed along the post-central gyrus. Remember our somatosensory map for touch and body position, in a very specific location. So mapping, basically, the organization, all these receptors and where the cabling goes, where it gets distributed matters a great deal for our, understanding of the location of the stimulus as well.